Welcome to the Deep Dive, the place where we turn complex scientific literature into actionable knowledge, especially tailored for you. Today, we're undertaking a really fascinating deep dive into a molecule that, and I don't think this is an overstatement, fundamentally rewired how neuroscientists think about motivation and desire. We're talking about PT-141, also known as bromolanotide. And this isn't your typical peripheral agent. It's an agent that targets the motivation center of the brain directly.
And that central first distinction is it's the absolute core of our mission today. It's everything.
Yeah.
We're looking at a synthetic heptapeptide, and our goal is to extract the critical insights from its research history. So we're going to try and understand its mechanism on the melanocortin system, trace its really surprising path of discovery, and maybe most importantly, dissect the massive technical challenges that arose from its first delivery format, the intranasal spray. For the curious learner, this molecule is just a perfect entry point into neurobehavioral science.
I think we need to cut straight to that defining central breakthrough. When we talk about PT-141 activating desire at the neural level, we're essentially discussing top-down regulation.
Right.
That this is the brain deciding rather than the body just reacting to localized physics.
It forces us to completely rethink the traditional model of arousal. It really does. PT-141 is such a powerful scientific tool because it demonstrates, unequivocally, that motivation and arousal can be orchestrated entirely by central brain circuitry. We're talking specifically about the dopamine pathways and circuits governing emotional salience, all before any peripheral physical response is even necessary.
So it's a literal demonstration that the brain leads and the body follows.
Exactly. The brain leads, the body follows. That's the takeaway.
Okay, let's unpack this from a technical standpoint. We are jumping into Section 1, PT-141's mechanism and identity. Before we dive into its function, what is PT-141 chemically? What does it even mean to call it a synthetic heptapeptide?
Right, good place to start. So technically, PT-141 is a synthetic peptide, meaning it's a short chain of amino acids, but synthesized in a lab, not by the body. The hepta part just means it's made up of 7 amino acid residues linked together.
So it's small.
Very small. And this structural identity is important because it tells us it's a relatively small, protein-like messenger. It was initially developed from some really intense research on what's called alpha MSH.
Alpha melanocyte stimulating hormone.
That's the one. The naturally occurring, slightly larger peptide cousin that regulates a whole host of core functions in the body.
And what specific part of the brain is it trying to talk to? Where is the target?
Its primary targets are the melanocortin receptors, specifically MC3R, and most pivotally for what we're talking about, for motivation, MC4R. And both of these receptors are highly, highly localized in the central nervous system.
So when you say central nervous system, we're not talking about the whole brain. We're talking about specific spots.
Very specific. We're really talking about key structures deep within the brain, particularly the hypothalamus and the reward-related regions like the paraventricular nucleus and the medial preoptic area. These areas are just foundational to motivation and all our survival drives.
Let's spend a moment to really isolate that idea. Central modulation versus peripheral action.
Yeah.
You mentioned the breakthrough is that it doesn't influence desire through circulation or a smooth muscle relaxation. So if it's not changing blood flow, what is the observable effect at the cellular level? What's actually happening to the neuron?
At the cellular level, it is a change in the electrical properties of neurons. It's that fundamental. When PT-141 binds to the MC4R receptor, it acts as an agonist. It initiates a signal. This binding fundamentally alters the neural firing patterns of the target neurons in the hypothalamus.
So it's making them more likely to fire.
Exactly. If you compare it to a purely peripheral agent, say one that increases nitric oxide signaling in localized tissue, PT-141 does the complete opposite. It starts the process by enhancing the neural readiness for behavior. The brain gets ready first.
Why is that distinction so critical for researchers? Why couldn't they just study desire using agents that boost peripheral responses? What was missing?
Because desire, motivation, subjective arousal. These are fundamentally psychological and emotional states. They happen in your head. Peripheral agents, like those used to treat erectile dysfunction, they're only effective if there is already pre-existing neural desire and activation.
So they're just helping the body catch up to the brain.
They facilitate the physical result of that excitement precisely. PT-141, on the other hand, allows researchers to bypass the need for external stimuli to some extent and instead look at the internal brain-driven networks. We can finally ask, how does the brain encode motivational drive? How does it assign emotional significance? It lets us study the motivational spark itself.
It sounds like the whole melanocortin system, this MC1R through MC5R family, is kind of master regulator for many survival needs. Is that why researchers talk about it as a system governing drive states?
That's precisely right. The melanocortin system is a foundational biological regulator of drive. It's not siloed. It doesn't just manage one isolated system. It manages fundamental survival behaviors. Hunger and satiety, so energy balance,
Right.
thermal regulation, social bonding, and yes, mating behavior and reward seeking. It's a totally interconnected circuit. For example, a great deal of the research on PT-141's mechanisms overlapped initially with appetite suppressants.
Oh, that's interesting.
Yeah, because MC4R is a crucial switch for both energy expenditure and hunger. It's a core system that governs whether you are motivated to seek something, food, a mate, warmth, or stay satiated.
And focusing in on PT-141's main target, the MC4R, why does this specific receptor serve as the key switch in the hypothalamus that specifically tilts circuits toward arousal?
Location, location, location. MC4R is highly concentrated in the medial preoptic area, or MPOA, of the hypothalamus. And this MPOA is recognized as probably the most critical integrating center for sexual motivation in the entire mammalian brain. So when PT-141 potently activates MC4R, it doesn't just cause random firing, it biases the activity of those MPOA neurons.
It pushes them in a specific direction.
It increases the probability that these circuits will fire and initiate the complex cascade required for motivational drive. And that includes making the individual more attentive and more receptive to potential sexual cues. It's tilting the internal balance towards seeking and engagement.
You know, for someone just getting into neurobehavioral science, PT-141 seems like an ideal case study because it just breaks down that old idea of compartmentalized biology.
It's the perfect teaching tool. It really is. Students realize very quickly that activating a relatively small set of receptors, the MC4Rs, doesn't just cause a simple physical reaction. It simultaneously impacts psychological states, emotion, attention, imagination, and behavioral drive. It beautifully illustrates this concept of neurobehavioral overlap, demonstrating that these core survival drives all share common molecular pathways in the brain.
So you can't really separate appetite from energy from reproductive drive at the level of the melanocortin receptor.
You can't. They're all talking to each other.
We mentioned that PT-141 was a refinement of an earlier analog, melanotan II. What was that process of refinement like? What was the technical design challenge they were trying to solve there?
The initial analogs, like melanocortin II, possessed the powerful central behavioral potency researchers were excited about, but they just weren't selective enough. They were a bit of a shotgun blast. Melanotan II hit multiple melanocortin receptors, and crucially, it was hitting MC1R, which is the receptor heavily involved in pigmentation pathways in the skin.
Ah, the tanning effect.
The tanning effect. So the technical challenge was to retain the behavioral potency, the MC4R activation, while eliminating, or at least dramatically reducing, the secondary effects on pigmentation.
So PT-141 was specifically engineered to be a cleaner, more selective tool for the brain.
Precisely. By subtly modifying the amino acid sequence of that 7-part chain, they were able to dramatically reduce its affinity for MC1R while maintaining, or in some ways even enhancing, its agonist activity at MC4R. And this refinement completely transformed the research landscape. It moved the molecule from a broad-spectrum analog to a selective research tool focused almost entirely on central behavioral circuits.
Which allows for much clearer cause and effect studies in motivation.
Exactly. Cleaner data.
Okay, let's trace this fascinating, messy and nonlinear path of discovery. We're moving into section 2, the nonlinear history of discovery. You said this started with pigmentation studies. How on earth did the research team jump from skin color to brain behavior?
It's the classic story of scientific serendipity, and it just highlights why observation is often the most important part of research. The initial research wasn't focused on behavior at all, not even a little bit. They were working on stable analogs of alpha MSH to see if they could modulate melanin production.
So basically, developing agents related to tanning or maybe skin protection.
Exactly that. They were running clinical trials, likely with human volunteers and definitely animal models, and they were just observing skin changes. That was the goal. But in the early melanotan II studies, researchers noticed these powerful and utterly unexpected behavioral responses that just kept cropping up, first in the animal models and then later, anecdotally, in the human participants.
And these weren't subtle effects.
Not at all. They were robust, observable changes in grooming, investigation, and mating behavior. The side effect was so prominent and so repeatable that it couldn't be ignored as some kind of mere anomaly.
That must have been a huge scientific pivot. I mean, to go from dermatology to neuroscience, how did they confirm that this behavioral effect was completely separable from the tanning effect?
That's when they did the chemical detective work. It's really clever. They took the melanotan, the second molecule, and started breaking it down into smaller fragments, testing each little piece for activity. And they successfully isolated the specific fragment, the key amino acid sequence that was responsible solely for these striking central behavioral effects, while largely dropping the activation of the MC1R pathways responsible for skin color.
And that fragment became PT-141.
That isolated fragment evolved into PT-141. It proved that the central action and the peripheral action were mediated by different structural features and could therefore be separated.
So once they had PT-141 isolated, what did the preclinical validation, the testing in rodents and primates, show that gave them such confidence in this central mechanism?
The preclinical studies provided the necessary anatomical proof. This was the key. Researchers would administer the new PT-141 peptide and then immediately look at the brain tissue. They used techniques like measuring CFOS expression.
Okay, for our listener, can you briefly explain what CFOS expression is and why it's relevant here?
Of course. So CFOS is an immediate early gene, and its protein product acts as a highly reliable marker for recent neuronal activity. Essentially, if a neuron has been actively firing over the last couple of hours, it will express high levels of CFOS protein. It's like leaving a little flag that says, I was just active.
A cellular
The
footprint.
cellular footprint, exactly. And the striking finding in the PT-141 animal studies was a massive increase in CFOS expression, specifically within the hypothalamic neurons in the MPOA, which are the very cells known to orchestrate motivational behavior.
That is compelling evidence. That's the smoking gun, proving that the chemical is talking directly to the intended neural circuit.
It was. This anatomical confirmation, you see strong arousal behaviors correlated with confirmed central neuronal firing without relying on any peripheral mechanism. That was revolutionary. It strongly suggested that the melanocortin pathway could be manipulated pharmacologically to study motivation and desire independently of traditional vascular pathways, which was a significant advance in the field.
So when they transitioned to human research, the goals were already clearly defined by this central mechanism. They weren't just looking for physical outcomes anymore.
Exactly. The human studies were fundamentally about assessing subjective and cognitive endpoints. They were looking for changes in desire, motivation, fantasy, the perceived emotional significance of intimacy. They were seeking evidence that the central mechanism they proved in animals translated to a cognitive and effective reorientation in humans.
And it did.
It did. The fact that the human subjective reports align so consistently with the mechanistic predictions from the preclinical work just confirmed the power of this top-down model.
And this is where the conversation about delivery begins. It's such a critical part of the story. Why did they opt for the intranasal spray format so early in the human trials? What was the hope?
The rationale was sound on paper, and it was highly appealing for a molecule designed to act centrally. The nasal mucosa is one of the quickest routes to achieve systemic circulation. I mean, it can rival injection speeds because of its high vascularity and relative permeability. So researchers hoped the spray would provide rapid systemic uptake and fast engagement of the neural pathways, leading to a quick functional onset, maybe within an hour or two, which would mimic the speed of a naturally occurring motivational state.
So the key lesson for the curious learner here is that the deepest insights often come from the most unexpected places. It's a cliche, but it seems true.
Absolutely. The entire field of central arousal research was redefined by a peptide that started its life aiming for skin pigmentation. It just teaches us that true scientific discovery is often messy, it's nonlinear, and it relies on researchers being open to observing and investigating unexpected phenomena that fall way outside the bounds of their initial hypothesis.
Let's move into section 3, because this is where the mechanism gets truly deep. We know PT-141 binds to MC4R in the hypothalamus, but let's trace that signal step by step. What happens inside the neuron at the moment of activation?
Okay, this is the critical cascade. When PT-131 docks onto the MC4R, which is a G protein coupled receptor, a GPCR, it triggers a complex intracellular signaling pathway. Specifically, it activates the GS protein subunit, which in turn stimulates an enzyme called adenyl cyclase. And this enzyme then rapidly produces large amounts of a crucial second messenger molecule, cyclic AMP or CAMP.
Why is CAMP so important? What is its role as a second messenger?
Well, think of the peptide, PT-141, as the first messenger talking to the cell surface from the outside. CMP is the molecule that carries that message deep inside the cell. It's the internal memo. Increased CAMP levels then activate another enzyme called protein kinase A, or PKA,
Okay.
and PKA acts like a master switch. It starts phosphorylating, just adding phosphate groups to very cellular proteins and ion channels. This changes the neuron's permeability to ions.
Which ultimately leads to a depolarized state, meaning the neuron is much more likely to fire an action potential.
You've got it. That's the chain of command.
So the end result of this whole CAMB-MPKA cascade is literally turning up the electrical activity in the hypothalamus. And that increased firing occurs most notably where? We said it before, but it's important.
In the medial preoptic area, the MPOA, that core motivational nucleus. And when those MPOA neurons start firing more vigorously, they don't just stay isolated. They project their signals out to many other regions, effectively broadcasting the message of initiate-seeking behavior.
So how does that signal flow into the broader reward system? This is where the motivational amplifier, dopamine, comes in, right?
That's the critical linkage. The MPOA has direct and indirect connections that influence the mesolimbic pathway, specifically the VTA, the ventral tegmental area, and its projection to the nucleus accumbens.
Which is universally recognized as the brain's reward center.
The reward center. And MC4R activation downstream stimulates increased dopamine release in this crucial region.
We often hear about dopamine and pleasure, but in this context, what is dopamine's function? Is it pleasure, or is it something more fundamental to motivation?
It's primarily motivational drive and salience, rather than just pleasure. That's a common misconception. Dopamine enhances the emotional significance or the salience of certain cues. So when you release dopamine here, it doesn't necessarily make the current moment more pleasurable, but it strongly motivates the individual to seek out, attend to, and engage with the relevant stimuli.
So in this case, erotic cues or potential partners?
Exactly. It's what drives that focused attention and prioritization. It turns a mild interest into a compelling motivational drive.
That explains the mental focus. But desire is inseparable from emotion. How does PT-141 activation influence emotional processing?
Right. This involves the limbic system, particularly the amygdala. The amygdala, which assigns emotional weight to stimuli and processes fear, novelty, reward. It becomes measurably more responsive during PT-141-induced activation. This heightened responsiveness means the brain assigns a stronger, more intense emotional weight to intimate cues. If desire is about feeling connected and interested, this mechanism is essentially turning up the emotional significance of those feelings.
I like that analogy of turning up the brightness on emotional salience.
Yeah.
So that suggests that we are focusing on the cues, but are we also reducing the distracting mental noise? Does this central action influence imagination and cognitive processing?
Absolutely. Functional brain imaging, fMRI, is key here. Researchers observed increased activity in regions associated with sexual imagination and fantasy, which confirms the cognitive activation. But perhaps the most interesting finding is the simultaneous decrease in activity in regions associated with self-monitoring, anxiety, or inhibitory cognitive processes.
The regions responsible for self-conscious or distracting inner critic thoughts.
That's it, exactly. Your inner critic gets quieter.
So PT-141 is working on a dual front. It's boosting the motivation circuits while simultaneously taking the foot off the mental brakes.
That's the elegant synthesis of the central mechanism. The net result is a highly optimized state where the individual is capable of more immersive mental engagement with imagery and fantasy without being immediately pulled out of that state by internal self-consciousness or distraction.
The mental stage is set for deep immersion.
Perfectly put.
Let's pull that thread back to the MPOA projections you mentioned earlier. How does this cascade firing MPOA neurons, dopamine release, heightened emotional salience, how does it physically manifest? How does the body follow?
So the MPOA acts as the command center, and one of its key outputs is to descending pathways, like the periaqueductal gray, or PAG, in the brainstem, which controls many core autonomic functions and motor patterns. When the MPOA is activated, it signals these lower brainstem centers to initiate the physiological events associated with arousal autonomic outflow, changes in heart rate, respiration, and then eventually the peripheral physical responses.
So this is the crucial step. The motivation and decision happen upstairs in the hypothalamus and the nucleus accumbens. The command to execute is sent downstairs.
That's the hierarchy. Brain first, body second.
We've established that PT-141 is this near-perfect model for central motivation. But Section 4 introduces a massive problem. How do you get this perfect little molecule into the brain predictably? Let's tackle the complexities of the intranasal spray delivery. Why did this seemingly great idea fail so spectacularly to deliver consistent results.
It's a tragic paradox in pharmacology, isn't it? You can have the perfect drug target, but if the delivery mechanism is unreliable, the research is, well, it's useless. The initial rationale for the intranasal spray was sound. Rapid onset, non-invasiveness, high vascularity of the nasal mucosa. It promised quick access to the systemic circulation and eventually the central nervous system. It looked great on paper.
But the sources are crystal clear. Bioavailability was unpredictable. How variable are we talking about here? Is it a small difference or a huge one?
It was profoundly inconsistent. It introduced massive noise into the research. Bioavailability, which is the fraction of the administered dose that actually reaches the systemic circulation, it varied by several fold, sometimes up to six-fold, even across individuals receiving the identical measured dose from the same device.
Wow, sixfold. For researchers, that is a complete nightmare because you can't be sure if a participant is experiencing no effect because the drug doesn't work,
Wow.
or simply because they absorbed a tiny fraction of the intended dose.
It makes the data almost uninterpretable.
What are the key technical factors influencing this absorption? This sounds far more complex than just spraying something up your nose.
It involves a whole host of highly dynamic variables. We can start with just basic anatomy. People's nasal passages are vastly different. Size, shape, internal topography, the curve of the septum. These differences dramatically alter the deposition pattern of the spray. So if the spray hits the highly absorptive, vascularized lower mucosa, absorption is high. Great. But if it hits the relatively non-absorptive septum or just drips down the back of the throat, absorption plummets.
And I imagine the technique of the user is also a major variable, which is incredibly hard to control for in a mass research setting.
Immensely difficult. The angle you hold it at, the force of the pump, the depth of insertion, whether the user sniffs hard or breathes normally. All these factors change the droplet size and the deposition depth. In a clinical trial, trying to standardize this physical technique across hundreds of participants proved to be nearly impossible, which guaranteed inconsistent mucosal exposure.
And let's not forget the biological barriers. You mentioned the health of the mucosa itself is constantly changing.
That's another huge source of day-to-day variability. I mean, think about factors like seasonal allergies, a common cold, a dry climate causing mucosal irritation.
Or even subtle hormonal changes.
Exactly. They can all change the permeability and thickness of the nasal lining. If the lining is congested or inflamed, absorption slows down dramatically and unpredictably. This makes cross-day comparisons highly questionable.
And on top of all that, there's the body's natural defense mechanism, which is actively working against the study.
That's the mucociliary clearance system. Your nasal cavity is lined with these tiny little hairs, cilia, that are constantly sweeping mucus and foreign particles toward the back of the throat to be swallowed or expelled. For a peptide, this means there is only a very short window, maybe 15 to 30 minutes, for absorption before the peptide is literally swept away and destroyed in the stomach.
So if the absorption rate varies, the amount cleared before being absorbed also varies widely.
You've got it. It's a race against time that some people win and some people lose with the same dose.
So if absorption is that variable, how does that translate into technical pharmacokinetic difficulties?
Yeah.
Does this affect the peak time of concentration?
It massively impacts the Tmax, or the time to maximum plasma concentration. With predictable delivery, like an injection, you have a very tight window for Tmax. You know when it's going to peak. With the intranasal spray, the Tmax was all over the map. For some participants, the peak plasma level, and thus the peak effect, was reached in an hour. For others, it might take 2 or 3 hours, or worse, never reach a therapeutic threshold at all.
And if researchers are timing their behavioral assessments based on a generalized Tmax, they're fundamentally mismeasuring the effect for a huge percentage of their participants.
They're missing the window. Or the window never opens. It created enormous difficulty.
And not just for the data.
Not just for the data, it was unsafe. High variability meant that some individuals, due to perfect anatomy and technique, would absorb an overly high dose, leading to magnified autonomic side effects. Others, absorbing too little, would appear non-responsive, potentially leading researchers to incorrectly conclude the drug was ineffective. The delivery method compromised the data integrity from every angle.
Given that the mechanism itself was so promising, this delivery issue eventually forced a change in focus, I assume.
Yes. The profound variability proved that the intranasal spray, at least in its early formulation, was not a viable clinical or robust research route. It just wasn't workable. This led research focus to shift to more predictable routes, primarily subcutaneous under the skin injection, which offers 100% bioavailability and highly predictable pharmacokinetics.
But the spray research remains an invaluable scientific case study.
It does. It's a perfect illustration of how the physical route of administration can make or break an otherwise successful drug concept. A cautionary tale for pharmacologists.
So the takeaway for any student reviewing pharmaceutical research is to never view the delivery route as a passive detail. It's not just how you take it.
Never. The delivery route is an active variable. It is a fundamental part of the scientific inquiry. If you can't guarantee a consistent dose reaches the target, all the brilliant biology and elegant mechanism in the world won't save the study.
Let's move on to Section 5 then. Human studies, neural signatures, and safety, focusing on the research outcomes once a more predictable delivery route was established, or at least in the high-response groups of those spray trials. What did human studies consistently reveal about the desired related changes?
Across multiple controlled settings, the findings regarding desire measures were consistent and, frankly, quite striking. Participants reported significant improvements in subjective desire and motivation, but crucially, they often reported a shift in the quality of their desire. They'd say they felt more mentally present, more curious, more open to intimacy, and less mentally resistant.
So it's less about a physical change and more about a mental reorientation toward engagement.
That's a great way to put it.
And that lines up perfectly with the idea we discussed of dampening the inner critic. We talked about fMRI data earlier. What is the robust neural signature we can observe that confirms these subjective reports?
The functional brain imaging provides remarkable confirmation. We see a robust signature of increased activity in the deep limbic structures, the nucleus accumbens, the MPOA, and the amygdala, when participants are shown erotic or intimate cues. Essentially, the brain's motivational centers, its emotional weighting centers, and its reward pathways are all lighting up.
And the decreased inhibition we discussed, how visible is that on the imaging?
We see a corresponding decreased activity in certain prefrontal regions associated with executive control and self-monitoring. This pattern suggests the brain is becoming highly optimized for focusing on the motivational cues, minimizing distraction, and suppressing that internal noise or self-conscious inhibitory processes.
The brain becomes receptive and hyper-focused on the rewarding stimuli.
Precisely.
I find the consistency of these findings incredibly validating for the researchers. It must have been a huge relief after the delivery mess.
It is rare and it's powerful. When you have neural data, behavioral observations, and subjective reports from participants all converging and all pointing back to the predicted mechanism, the melanocortin pathway, it elevates the PT-141 research from a simple drug study to a foundational model for understanding motivational neurobiology.
Now let's talk safety and tolerability. When you push such a powerful central switch, there are always going to be side effects. What adverse effects were consistently and reliably observed?
The most common and really unavoidable adverse effect observed across trials, regardless of the delivery route, is nausea. Following that, flushing, headache, and transient increases in blood pressure are also frequently reported autonomic effects.
And you mentioned something important. These effects tend to be more intense with initial exposure, and then they often decrease with repeated administration in research protocols.
That's right. There seems to be an adaptation or tolerance that develops.
Let's focus on the nausea for a second, as it seems the most counterintuitive. Why does a peptide that acts in the brain's motivational centers cause gastrointestinal discomfort?
It's an excellent question that goes right back to the interconnectedness of the melanocortin system. The MC4R receptor system doesn't just influence motivation, it also influences the autonomic nervous system, the system governing all our involuntary functions. This includes the central regulation of gastrointestinal motility and visceral sensation, often mediated through vagal nerve connections in the brainstem.
Oh.
So when the MC4R system is suddenly and powerfully activated by PT-141, it disrupts this established autonomic balance. This sudden shift results in a central signaling that the gastrointestinal system interprets as discomfort, and that manifests as nausea. It's a clear biological signature of the central nervous system's command over visceral functions.
That connection explains why the side effects aren't isolated. It shows that central activation inevitably touches peripheral autonomic systems.
Exactly. The flushing and the temporary, mild increases in blood pressure are other examples of that autonomic intertwining. The central nervous system is managing cardiovascular tone. When you activate a major central survival pathway like the melanocortin system, you are influencing the fight-or-flight balance, leading to these transient, sympathetic-driven effects.
And finally, what about the pigmentation issue? Did the refinement of PT-141 completely eliminate the skin darkening effect from melanotan II?
Not entirely. While PT-141 was specifically designed to be far more selective for MC4R, melanocortin receptors are still a related family. There's crosstalk. There remains a mild, low-affinity interaction with the MC1R receptor, which mediates melanin production in the skin. Therefore, during extended or frequent administration in research settings, a mild, localized, or sometimes generalized darkening of the skin is still an observable phenomenon that researchers must track and account for.
Which is a crucial detail because it confirms the peptide is still acting on the broader family of receptors, even if it is highly specialized.
It's a reminder of its origins.
So that means the concluding safety insight for anyone engaging with this research is that high specificity doesn't mean zero crosstalk.
That's the key lesson. Because PT-141 is such a powerful central actuator manipulating core survival drives, the systemic consequences are significant. Any researcher needs to approach this with careful screening, constant monitoring, and a high degree of control to manage these predictable autonomic shifts and ensure the safety of the research participants.
We've now covered a profound amount of detail, from the molecular sequence all the way to the clinical challenges. As we wrap up this deep dive, let's zoom out and look at the big picture. What does PT-141 fundamentally teach us about the neuroscience of motivation?
The central message, I think, is that PT-141 provides researchers with a truly unique scientific window, giving us direct access to the brain's core motivational machinery. It essentially confirms that desire is not primarily a physical reflex, but an emotional, cognitive, and attention-driven state dictated by this powerful melanocortin pathway. It's a template for understanding central drive.
So if you are a learner coming to this deep dive for that single most valuable nugget of knowledge, what should you walk away remembering?
That arousal and motivational engagement in general begins in the brain. It starts there. If you understand the specific pathways, the MPOA, the dopamine release in the nucleus accumbens, and the emotional weighting in the amygdala, then all the subsequent behavioral and physiological outcomes become predictable. The neurobiology of motivation is a top-down system.
Building on this foundation, what are the exciting future research directions that PT-141's history has opened up? Where do we go from here?
Several areas are thriving. One is in advanced pharmacokinetics, attempting to redeem the delivery concept by refining intranasal absorption. They're looking at advanced excipients or novel devices to reduce that crippling variability we discussed.
So trying to fix the spray.
Trying to fix the spray, exactly. Another is highly detailed, dynamic neuroimaging linking precise, known plasma concentrations of the peptide to temporal shifts in brain activity, real-time mapping. But the most exciting field, I think, is cross-domain motivation research. Since the MC4R pathway regulates both desire and energy balance, researchers are using PT-141's mechanism as a tool to investigate how generalized drive states like the motivation to eat or the lack of motivation seen in some types of anorexia share the same molecular switch as motivational desire.
So the template is universal.
The template is universal, drive is drive.
That gives us plenty to mull over. I want to leave our listener with one final provocative thought that ties back to the sheer power of this central switch. Since PT-141 can modulate neural circuits to increase motivational drive for specific cues, what does this tell us about the generalized mechanisms of motivation and reward seeking that are entirely unrelated to desire?
That's the fundamental question for the next generation of neuroscience, isn't it? If we can pharmacologically enhance the central salience of one set of cues, what are the implications for non-survival related motivations? What could this teach us about sustaining focus, about managing compulsive seeking behaviors, or even the intense reward seeking necessary for complex cognitive acts like creative innovation?
Or addiction.
PT-141 shows us the master blueprint for drive, regardless of the ultimate target. And that foundational knowledge is incredibly powerful.
It certainly provides a framework for tackling some of the biggest questions in human behavior. Thank you for walking us through this incredibly detailed and complex research model.
My pleasure. This field is only going to grow more detailed, and understanding these central mechanisms is crucial for anyone following the cutting edge of behavioral science.
And finally, we must deliver this crucial research disclaimer. The peptides and products discussed in this deep dive are strictly limited to research purposes only and are not intended for human consumption. This distinction between research tools and clinical use is fundamental to the scientific protocol that governs this area of study. Thank you for joining us for another deep dive. Make sure to subscribe so you don't miss our next analysis of crucial research. We'll see you soon.